Good day everyone, this is Lian Mae R Ayade, and I will be discussing to you about Wilson's disease. So Wilson's disease is a rare inherited disorder that causes copper to accumulate in your liver, brain, and other vital organs. Most people with Wilson's disease are diagnosed between ages 5 and 35, but it can affect younger and older people. This disease bears the name of the British physician Samuel Alexander Kinnear Wilson, 1878 to 1937, a neurologist who described the condition, including the pathological changes in the brain and liver, in 1912. Wilson's disease is caused by a mutation in the Wilson's disease protein ATP7B gene. This protein transports excess copper into bile, where it is excreted in waste products. Wilson's disease is inherited as an autosomal recessive trait, which means that to develop the disease, you must inherit one copy of the defective gene from each parent. If you receive only one abnormal gene, you won't become ill yourself, but you're a carrier and can pass the gene to your children. ATP7B gene provides instructions making a protein called copper-transporting ATPase2. This protein is part of the P-type ATPase family, a group of proteins that transports metals into and out of the cells by using energy stored in the molecule adenosine triphosphate. Copper-transporting ATPase2 is found primarily in liver, with smaller amounts in the kidneys and brain. It plays a role in the transport of copper from the liver to other parts of the body. Copper-transporting ATPase2 is also important for the removal of excess copper from the body. Within liver cells, copper-transporting ATPase2 is found in a structure called the Golgi apparatus, which modifies newly produced enzymes and other proteins. Here, copper-transporting ATPase2 supplies copper to a protein called ceruloplasmin, which transports copper to other parts of the body via the blood. If copper levels in the liver get too high, copper-transporting ATPase2 leaves the Golgi and transfers copper to small sacs for elimination through bile. Bile is a substance produced by the liver that is important for digestion and the removal of waste products. Copper is an important part of certain enzymes that maintain normal cell functions. Copper plays a key role in the development of healthy nerves, bones, collagen, and skin pigment melanin. Normally, copper is absorbed from your food and excess is excreted through a substance produced in your liver. Copper is needed by the body for a number of functions, predominantly as a cofactor for a number of enzymes such as ceruloplasmin, cytochrome C oxidase, dopamine B hydroxylase, superoxide dismutase, and tyrosinase. Wilson's disease is found worldwide with an estimated prevalence of 1 case per 30,000 live births in most populations, although data from population screening by molecular sequencing in the United Kingdom suggests a potentially higher prevalence, perhaps as frequent as 1 case in 1721. Assuming a prevalence of 1 in 10,000 to 30,000, approximately 1 person in 90 carries an abnormal copy of the ATP7B gene. However, in some isolated populations, the prevalence is higher. One of the highest reported prevalence was on a small mountain on the island of Crete where Wilson's disease was diagnosed. Some studies suggest that men and women are equally affected by Wilson's disease, though women are more likely than men to develop acute liver failure due to Wilson's disease. The main site of copper accumulation are the liver and the brain, and consequently liver disease and neuropsychiatric symptoms are the main features that lead to diagnosis. People with liver problems tend to come to medical attention earlier, generally as children or teenagers, than those with neurologic and psychiatric symptoms who tend to be in their 20s or older. Some are identified only because relatives have been diagnosed with this disease. Many of these, when tested, turn out to have been experiencing symptoms of the condition but not have been received diagnosis. Liver disease are the hepatic encephalopathy, portal hypertension, chronic active hepatitis, and hepatocellular carcinoma. Neuropsychiatric symptoms are Parkinsonism, ataxia, dystonia, seizures, migraine, frontal lobe disorder, and subcortical dementia. Behavioral changes are depression, anxiety disorder, and psychosis. Other organs may be affected, such as eyes, or the case of Fleischer's, kidneys, renal tubular acidosis, heart, cardiomyopathy, hormones, hypoparathyroidism, infertility, and recurrent miscarriage. There is no totally reliable test for Wilson's disease, but levels of ceruloplasmin and copper in the blood, as well as the amount of the copper excreted in urine during a 24-hour period, are together used to form an impression of the amount of copper in the body. The gold standard of most ideal test is liver biopsy. Diet. In general, a diet low in copper-containing foods is recommended with the avoidance of mushrooms, nuts, chocolate, dried fruit, liver, sesame seeds, and sesame oil and shellfish. Medication. Medical treatments are available for Wilson's disease. Some increase the removal of copper from the body, while others prevents the absorption of copper from the diet. Penicillamine is the first treatment used. This binds copper and leads to accretion of copper in the urine. Physical and occupational therapies. Physiotherapy and occupational therapy are beneficial for patients with neurologic form of disease. The copper chelating treatment may take up to 6 months to start working, and these therapies can assist in coping with ataxia, dystonia, and tremors, as well as preventing the development of contractures that can result from dystonia. Transplantation. Liver transplantation is an effective cure for Wilson's disease, but is used only in particular scenarios because of the risks and complications associated with the procedure. It is used mainly in people with fulminant liver failure who fail to respond to medical treatment, or in those with advanced chronic liver disease. Liver transplantation is avoided in severe neuropsychiatric illness in which its benefit has not been demonstrated.